Alright, let's begin our investigation by looking into the effect of varying light intensity on the rate of photosynthesis in submerged aquatic plants. So, this investigation is all about understanding how changing the light intensity affects the rate of photosynthesis in submerged aquatic plants. Coming to the objective of the experiment, the purpose is to find out how different levels of light influence the plant's ability to photosynthesize. We will measure the amount of oxygen produced by the plant and also observe color changes in a carbon dioxide indicator solution, which tells us how much carbon dioxide is being used up. Both of these give us clear evidence of the rate at which photosynthesis is happening. This also reminds us that while light is important, it is just one of many environmental conditions needed for photosynthesis to take place effectively. Now let me quickly explain how we will set up our experiment. We will place an aquatic plant, like a lodi, in a beaker filled with water and hydrogen carbonate indicator solution. This solution provides carbon dioxide and also changes color to show how much is being used during photosynthesis. The beaker will sit in a water bath to keep the temperature steady, and we will use a thermometer to monitor it. Above the plant, we will place a gas collection tube to measure how much oxygen the plant produces. A lamp will be used as the light source, and we will use a ruler to change and measure the distance between the lamp and the plant. This helps us control the light intensity. This setup will let us measure the effect of different light intensities on the rate of photosynthesis while keeping other conditions constant. So, next, moving on to the materials needed for this investigation. We will need the following: a submerged aquatic plant, such as a lodi or kabomba, a hydrogen carbonate indicator solution, which provides carbon dioxide and changes color based on carbon dioxide levels, a beaker or test tube to hold the plant and the solution, a lamp or a consistent light source to change light intensity, a gas syringe or an inverted measuring cylinder to collect and measure oxygen, a stopwatch to time the experiment, a thermometer to ensure temperature stays the same, and a ruler to measure the distance between the lamp and the plant. Then, coming on to the preparation part, we will first prepare a carbon dioxide solution by dissolving sodium bicarbonate in water. This gives the plant a constant supply of carbon dioxide. A 1% solution is commonly used so that carbon dioxide remains the same in all parts of the experiment. This way, we can focus only on the changes in light intensity. Now let's move on to the experimental setup. We start by placing the aquatic plant into a beaker or test tube filled with the sodium bicarbonate solution. Then we place a lamp at a starting distance, for example 10 centimeters from the plant. This gives us the highest light intensity to begin with. We use a thermometer to keep the temperature steady at around 20 to 25 degrees Celsius. And finally, we set up our gas collection method using either a gas syringe or an inverted cylinder to collect oxygen from the plant. Coming on to the method of changing light intensity. We do this by changing the distance between the light source and the plant. The closer the light, the more intense it is. As we move the lamp farther away, the light gets weaker. We can use common distances such as 10 centimeters, 20 centimeters, 30 centimeters, and 40 centimeters. This change in light follows a scientific rule called the inverse square law, which tells us that light intensity decreases sharply as distance increases. Now let's go step by step through the procedure. First, place the plant in the prepared solution. Next, position the lamp at 10 centimeters and start the stopwatch. Allow the plant to photosynthesize for a fixed time, let's say 10 minutes. During this time, measure how much oxygen is produced using the gas syringe or by collecting gas in the inverted cylinder. At the same time, observe any color change in the hydrogen carbonate indicator. This will shift from yellow, which shows high carbon dioxide, to green or blue, showing that the plant is using up carbon dioxide. Once this is done, move the lamp to 20 centimeters and repeat the same steps. Then again at 30 and 40 centimeters. Be sure to repeat each distance multiple times to make sure the results are accurate and reliable. Now, talking about the observations and what to expect. Now let's discuss what we expect to see. At 10 centimeters, where the light intensity is high, the plant should produce more oxygen. The indicator will likely change to green or blue quickly, showing fast carbon dioxide consumption and active photosynthesis. At 20 centimeters, the rate should decrease. You'll see less oxygen, and the color will change more slowly. At 30 centimeters, the oxygen will be even less, and the color might only change slightly. And at 40 centimeters, where light is very weak, the plant might produce no oxygen at all, and the indicator could remain yellow, meaning very little or no photosynthesis has occurred. Now coming to the results and what they tell us. The experiment shows that light intensity has a direct effect on the rate of photosynthesis. The closer the lamp, the more oxygen is produced, which tells us that photosynthesis is happening faster. Why is this? Because light is the energy source that powers photosynthesis. The more light the plant receives, the more energy it can use to carry out this process. So, there is a clear link between light intensity and photosynthesis rate. However, Even if we keep increasing the light, there will come a point where photosynthesis stops increasing. This is known as the plateau effect. At this point, something else becomes the limiting factor. It could be the carbon dioxide level, or the temperature, or even the plant's internal systems. In our case, we kept the carbon dioxide constant. But in natural conditions, if carbon dioxide is low, then increasing light alone won't help. The plant simply won't have what it needs to continue photosynthesizing at a higher rate. This idea is very important and teaches us that photosynthesis depends on several factors working together, not just light. Now to sum it all up, this experiment confirms that light intensity plays a major role in the rate of photosynthesis. The closer the light, the more oxygen the plant produces. The farther it is, the less photosynthesis occurs. However, the experiment also shows that light is not the only factor. To achieve optimal photosynthesis, plants also need a suitable temperature, enough carbon dioxide, and other ideal environmental conditions. So, to conclude everything, this investigation is a simple yet powerful way to show the relationship between light and photosynthesis. By moving the lamp and observing how the plant responds, we can clearly see how light energy drives this essential life process. It also helps us understand the concept of limiting factors, which is very important in plant biology. Overall, this is a well-planned experiment that helps us understand how plants depend on light to produce food and release oxygen.